It's my pleasure to be here tonight and to have the chance to tell you a little bit about a very complex area of cell biology that has at its underpinnings, in its basis, a very simple kind of solution. My origins as a scientist were in physics, especially theoretical physics. And I have always looked at the world in ways in which to try to simplify. And so as I became a scientist, I moved from physics into medicine and then into biology and biological research. But I always somehow retained the idea that even the very complicated processes of life have at their basis some simple underlying physical principles. And I've sought very hard to try to establish those in certain cases. Now, in recent years, my laboratory is very fortunate to have a deep collaboration with this institution, specifically with the Department of Statistical Physics, and specifically within that department with Eric Perez and with Professor Ponce. And in fact, we have students and fellows who we share and who sometimes go back and forth between New Haven, which is in Connecticut, which you probably have no idea where that is. It's near New York. And back and forth. And so our collaboration is a very active international collaboration, and I'm very enthusiastic about finding ways to only make that deeper. Several of the people actually I see in the 1, 2, 3, 4, 5, about the 10th row. Maybe you can raise your hands who are part of our team. Okay, so if you have any really tough questions, you now know who to address them to. The title that Fred chose for tonight's talk, it's a slightly different title that I chose, it was the Federal Express System of the Cell. And since some of you are non-scientists, I assume, it's a very open evening, could I ask, just to understand a little bit, could you raise your hand if you are not at École Normale? If you are someone from the real world. Thank you. And those of you, proudly, thank you for coming. And who of you are scientists at École Normale with PhD degrees or PhD students? Okay, good. And are any of you students, undergraduate students? I'm not sure what the exact term is here. Students in the 4-year program at École Normale. Okay, very good. So that's helpful to know. So I might just say at the very beginning that for those of you who are not scientists, that the cell is organized, the cell is a very complex environment that has many locations within it. Each of these locations is we call a compartment. And each compartment is surrounded by a little wall. That wall is called a membrane. These walled off compartments exist simultaneously in the cell with each other. There are many names for these compartments. It doesn't really matter what they're called, but let's call them A, B, and C, and D, and so on. There are different compartments. They exist within the cell as a whole, which itself is walled off by a membrane that separates the cell from all the other cells, and from the blood, and from everything else in the body. So the wall, or the cell membrane, defines the cell. Now, the cell makes the working parts, it makes its own working parts every time it grows and divides, and these are called proteins. They are the catalysts that carry out and facilitate the chemical reactions that occur within the cell. Now, each of the compartments has a special role. A, B, C, they do different things. In order to do different things, A has one set of proteins because these are the workers that do the jobs. B has a different set of proteins than A, C a different set again, and so on. The interesting problem is that all proteins are made by a special compartment in the cell called the ribosome. And so all of these proteins start out together. Even though some of them have to go over here to A, others have to go over here to B, still others to C. Does that make sense? And if they got mixed up during this process of transport within the cell, or sorting within the cell, if they got mixed up, A, B, and C would no longer have their unique functions and the cell would die. So this is really a vital process for the cell. Now, the cell has a solution to this process. The cell has within it a delivery system. And it's that delivery system that we've come to understand in rather precise molecular terms, and I will explain some of that, just some of it to you tonight. Now, when you have cargo, like the proteins that need to go to A or the proteins that need to go to B, they're cargo. They need to be carried. And in the real world, when you have cargo, what do we use if we want to get cargo around Paris? We'll use a delivery truck, right? Now in New York, it'll be a big truck because we have big, wide streets. But here, it'll be a small truck. And let us say the truck is a Federal Express or a DHL truck because you want it to get there quickly. So what does the truck have to do? The truck has to first go to a place where it loads the cargo, right? Then the truck has to find its way to the destination where the cargo goes. And the truck has to open its door and deliver the cargo, right? It's just that simple. Now the cell has perhaps 24 types of cargo trucks. They're very tiny, okay? They're nanostructures. They are actually about 500 angstrom units in size. What does that mean? The smallest atom in nature is the hydrogen atom, okay? These little cargo trucks are only 500 or so hydrogen atoms big. That's how small they are. And at any one moment in each of our cells in our body, every single one, we have perhaps 100,000 of these delivery trucks. Now these delivery trucks actually have a transient existence. They form where the cargo is loaded, and they are consumed as they deliver the cargo. So they live for a very short time, from seconds to a minute each. So picture 100,000 of these in every cell, as microscopic as that is, all of them forming constantly and delivering constantly. And each one must load with the right type of cargo, let's say cargo for A, cargo for B, cargo for C, and each one needs to deliver. If you're carrying the A cargo, you go to compartment A. You never go to compartment B. So there's something very fundamental that needs to be understood, because in order for a cell to grow and divide, it has to make another set of copies of all the compartments that I told you about at the very beginning. And it has to do it with great precision. Okay? So the result of all this then is that the cargo needs to be distributed by a process of transport similar to a delivery truck, and it needs to be loaded and unloaded. So that's the overall concept. And it's sort of actually kind of simple. Now, that basic concept came from this scientist, George Palade, in the 1960s. Now, Professor Palade, who received the Nobel Prize in 1974 for discovering the fact that there are transport pathways in the cell, actually, he's also my predecessor as the director of the department at Yale. So there's a certain nice history in this field. Now, what Palade did was he studied a certain type of cargo which is secreted from the cell. So I've told you that some cargoes are delivered within the cell, but other cargoes are actually released from the cell into the blood. We call that secretion. And I know everybody in this room is familiar with this because we've all heard of the hormone insulin. And insulin is a hormone that's secreted into the blood, and it acts on cells throughout the body to control their uptake of sugar, and that's how we control blood sugar. And so I think you can understand that the secretion of insulin is a very important process. And if it's imbalanced, our bodies no longer handle sugar and fat and also amino acids, other metabolites correctly. And we're then prone to diabetes, or we're prone to obesity, or both. Okay? So these are very important processes to keep in balance. Now, insulin and similar secretory proteins, as Palade discovered, are made within the cell. This is an example of a membrane-bound compartment. I'll call it A. It's actually technically called the endoplasmic reticulum, or ER. And the ER has attached to it these little particles that, and this is, by the way, the wall that I was telling you about that surrounds it, the membrane. Those little particles are called ribosomes. And these are the little molecular machines that make the new proteins. And they introduce these proteins into the inside of this organelle called the endoplasmic reticulum. And so what is introduced into that organelle is a mixture of all of the proteins that are going to go to A, to B, to C. So they actually are all mixed together, and then they need to be separated from each other. Cargo A, cargo B, cargo C, and each one somehow dispatched to the right location. So Palade discovered this basic organization, and in the case of proteins that are released or secreted from the cells, he discovered that these proteins pass through an intermediate set of compartments called the Golgi apparatus, named after Camillo Golgi, who actually received the medicine prize from a Nobel Prize in 1906, which I think was the first one. So again, a long history in this field. And they are then transported into what are called secretory storage vesicles that contain, at this stage, only the cargo that's going to be secreted, removed from all of the other cargo that goes to other places in the cell. And then in a moment, I'll show you how this cargo gets released from the cell. In other words, how the delivery occurs by a process that is technically called membrane fusion. So Palade discovered this, and he also noticed in the electron microscope, he was actually among the very first biologists to use the electron microscope in biology, starting in the early 1950s, late 1940s. As a result, he could see tiny particles like the ribosome in the cell that nobody had seen before. And he brilliantly interpreted these micrographs. He saw this is an example, a cartoon of the vesicle or the delivery truck that I was telling you about. So in terms of scale, this is, for the scientists in the room, this is about 50 nanometers in size. And this is an example, and you'll see many, of these carriers or cargo trucks that are technically termed vesicles. And you'll see that they have a membrane surrounding them very much like the membrane here. And to foreshadow a little bit, what Palade concluded was that these vesicles, or these cargo containers, I will now refer to them as vesicles rather than cargo containers or trucks. They form by a process of budding or blebbing off from a membrane. So this compartment, the endoplasmic reticulum, gives rise to this vesicle as the vesicle pinches off and forms its own little separate compartment. In so doing, it carries certain proteins, certain cargo, on the inside that it obtained during the budding process. And now, as it travels over to the next compartment, it can deliver this cargo by a process that's called membrane fusion. Membrane fusion is the opposite of membrane budding. Here, the vesicle forms as the wall of this compartment is shaped into a smaller spherical particle, the delivery truck. Now, this particle joins, this vesicle joins and binds to and joins with and actually merges its wall with the target compartment. Okay? And in so doing, it delivers its cargo. So this is what Palade taught us. And in the years since, we've learned that there are many related pathways that carry the cargo to the various other destinations in the cell, as distinct from just the secretory cargo. Now, the next major step in this field was taken by Günter Blobel at Rockefeller University, who received the Nobel Prize in 1999. And what he solved was the problem of how these proteins that go to the endoplasmic reticulum, how they know to go there. Because many of you know that there are other places in the cell that receive proteins by other pathways. So, for example, the mitochondrion receives its proteins from ribosomes via the cytosol. And proteins that remain in the cell sap or cytosol are actually made by ribosomes that are not bound to the endoplasmic reticulum membrane. They are basically the same particle, but they're localized differently. And what Blobel discovered is that each cargo molecule has a special zip code. So you see the analogy to Federal Express is very strong, because when you send a Federal Express shipment, you get a tracking number, right? So the cargo is you first determine, is this cargo A or B or C, and you barcode it. And that barcode is actually built into the sequence of amino acids that comprises the protein. Proteins A have a different signal sequence. Proteins B have a different signal sequence. All proteins B have the same signal sequence as each other. All of A have the same as each other, but A's signal differs from B's signal. It's a little more complicated than that, but not much, actually. That's how neat the cells are designed, okay? So the result of that, and what Blobel discovered, is the existence of these zip codes, as he called them, or signal sequences, that act like tracking numbers, essentially. And so when a cargo is packaged into a vesicle, there are receptors in the walls of these vesicles that actually bind to the signal sequence and concentrate these proteins here so that they are packaged. Cargo A gets into this vesicle, and cargoes B and C, which lack the ability to bind to the forming wall of the vesicle, they are not packaged. It's, again, a very simple idea. The biochemistry is complicated. But that is what Professor Blobel discovered. So what remained to be discovered is how these vesicles form and how they deliver their cargo. Okay? Now, I sort of gave you the answer to how they form. They form by budding. But how at a molecular level do they form? How at a molecular level do they select their cargo? And especially the most difficult part of the whole thing is when you have a dozen or more different types of vesicles in the cytoplasm, all mixed up, how does this one know to join and fuse over there, and this one know to join and fuse over there? And by the way, what makes the membranes fuse? Now, I want to take a moment and remind you what a membrane is made of. Okay? The walls of the cell and the walls of these organelles are made of two layers of fatty substances called lipids. Now, all of you are familiar with fatty lipids because you wash your hands with soap. Okay? I hope. And soap forms a so-called monolayer of a soap molecule is actually a breakdown product of the lipid constituent of these membranes. And it forms, as you know, a bubble. Okay? And we've all played with soap bubbles, and we know that they can bleb off of each other, and we also know that they can join together and merge and coalesce. That's what we mean by fusion. And I'll have more to say about that now, so in a moment. So these membranes that surround the cell, and actually, could we turn off the lights for these microscope pictures? Is that possible? I think it would be helpful. These, thanks, Fred. These membranes have a kind of liquid-like quality. So they're not like brick walls. They're deformable. They're fluid, but deformable. Well, just do the best you can. So the process of membrane fusion is needed for a whole variety of physiology. As I've already told you, it's needed for the distribution of proteins among the compartments in the cell. And this is actually an electron microscope image of the endoplasmic reticulum. I'm hoping you can see the little black particles. Those are the ribosomes. This is a vesicle that's forming. This is actually from Professor Pilati's original collection, which I got access to when I took over the department at Yale. And this was actually written in his hand. So it's actually of some historical interest. And these are the little containers. You see how many of them there are? Okay, these are the vesicles. This is the Golgi apparatus. And this is an example of a vesicle that stores a secretion. Okay? Now, the cell growth and division are just one kind of pathway that utilizes this. In addition, I've already mentioned the secretion of insulin, and thank you, Fred, and all the hormones persistence pays. And there's another example, and there are so many examples of the importance of this pathway, but the one that I'll come to in the latter part of the lecture, and the one that is very important to all of us, is the brain. Because there's a form of secretion in the brain. and that is the release of neurotransmitters. So when one nerve cell talks to another, it does so at a connection between the two to make a circuit, and that connection is called a synapse. And at the synapse, neurotransmitters are stored within the cell, and they are released. And they are released when the electrical signal comes down to the end of the wire, or axon, and it then triggers the release of this chemical signal that diffuses across the gap that connects the two nerve cells, and then stimulates the next nerve cell to fire electrically. So electricity is converted to chemistry and back to electricity every time you pass information in the brain. That has to happen very, very quickly, or else we are very, very stupid. Okay? So how quickly? Within 1/1000th of a second. That's how fast it is. So at the end of every nerve, this is a nerve that happens to be synapsing on a muscle. So this is the outer wall of the nerve blown up to a huge magnification. A nerve might go from here, from the spinal cord to the muscle. Here it touches the muscle. And we're looking right at this area right here at an enormous magnification, about 100,000 power. Okay? And these are the vesicles, synaptic vesicles they're called, that are at the synapse, and each one contains perhaps 20 or 30,000 neurotransmitter molecules. And this, when the nerve is stimulated, these vesicles, synaptic vesicles, fuse now with the outer membrane of the nerve cell, releasing their stored neurotransmitter into the space or the synaptic cleft, where they can now diffuse across the synaptic cleft and stimulate, in this case, the muscle to contract. In other cases in the brain, of course, it's not a muscle. We like not to have muscles in the brain. We have nerves, and that causes nerves to fire. So you can see that the delivery process of membrane fusion is really vital for a whole range of endocrine physiology, exocrine physiology, and as well as neurophysiology. And that imbalances in these pathways actually are very much at the heart of diseases like cancer, immune disorders, endocrine disorders, and even neurodegenerative diseases. So how does this delivery process work? So my story begins at a very young age. That's actually me. Where am I? Right here. And I'm assembling a rocket to launch. I'm in that picture, 12 years old. And the reason I mention this is that Fred wanted a little bit of a historical or a personal thing here. So I mentioned my interest in physics. I grew up in the 1960s, and that was the era of Sputnik and the space programs. And in that era, if you were interested in science, more than likely you were interested in physics, because that's what we always heard about, between the nuclear bomb, nuclear power, and the space program, and the enormous progress with transistors and electronics and then computers. Really, most young scientists were fascinated with physics, and I was no exception. And because of that orientation, I did, and still do, approach the world in the same conceptual framework as a physicist. So when I took on the problem of how membrane fusion occurs and how cargo transport occurs in the cell, like a physicist, physicists always look for universal laws that can explain all related processes on a common basis. And they always look for the simplest explanation for all the facts. Now, all of these processes are so different, each one from the other, that my colleagues, all of them, assumed that there would be some different mechanism for each of them. If I take you back to the late 1970s, when I started working on this problem as a young assistant professor at Stanford University, there was one group of people working on this, another group of physiologists or doctors working on this, and yet still another on this. And each one was absolutely convinced that they were different from the other and that there must be a different mechanism, because neurotransmitters are released under entirely different conditions than insulin, okay, or pancreatic enzymes, or in which cells divide. So there must be different mechanisms. But I sort of took the idea that there's maybe a common mechanism, and I tried to find the simplest system that I could think of that could reveal the underlying mechanisms. And in particular, I proceeded with the simplest possible hypothesis. Now, there were two ways to look at this problem back then. The first was the way that almost everybody else looked at it, and the second was the way that I looked at it, which is probably why I'm here giving this lecture tonight, and they are not. Okay? And there's a lesson in this, which is it pays to have your own way of thinking. Okay? And just because people tell you that what you want to do is stupid, and they did, in my case, the most famous people in the field, it doesn't mean that it actually was. Okay? Probably statistically they would be right 99% of the time, okay? But sometimes not, okay? And I try to remember that myself when I offer advice. You'll notice you've seen this micrograph now several times, but now I want to show it to you from a different point of view. You see how these vesicles forming from the endoplasmic reticulum are clustered next to the Golgi. These vesicles, in fact, are going into the Golgi. It is as though they are being fed directly from the endoplasmic reticulum in the Golgi. And the cell is highly organized in 3 dimensions within the cytoplasm. This happens over and over again. And so the idea that every electron microscopist had was that it must be absolutely critical that the endoplasmic reticulum and the Golgi are next to each other, because these vesicles are being fed directly into the Golgi. Okay? I took a different point of view, much more like a chemist or a physicist would, and thought, well, maybe that's true to some extent, but there must be some underlying inherent chemical specificity. Maybe each of these vesicles has some feature on its surface that is recognized by the target membrane, let's say A versus B, and that a partnering of proteins as yet undiscovered, protein machinery needed to cause the fusion process occurs. And so that if you take this way of thinking, that there's inherent specificity and that the anatomy emerges as an emergent property from the underlying specificity rather than the other way around, if you take that point of view, then you have a remarkably simple hypothesis. And that simple hypothesis is that vesicle traffic, which is the term for this delivery process and the transport process, can take place in a cell-free extract. Because if this is right, I should be able to disrupt the cell by homogenization, literally disrupt it, break the walls of the cells, and separate these organelles so that they're free-floating in solution. And if there is inherent chemical specificity, these vesicles should not know the difference. They should form as they would, as if they were in a living cell, if you get the right kinds of conditions, and they should then go on to deliver to their cargo with specificity as if the cell were still alive. Okay? Well, this is the basic tenet of enzymology or biochemistry. And it is a tenet that has been proven true over and over again in the 100 years since fermentation was first reproduced. Metabolism of glucose to make alcohol, essentially, was first produced in cell extracts of yeast by the German biochemist, the early biochemist, the Buchner brothers. And over again, biochemists have found that more and more complex processes can be reproduced in cell extracts. Now, the reason that's so important is that it provides the means to establish the molecular basis of this process. Because if you can reproduce the process in a test tube, you can then apply tools of chemistry, biochemistry, physical chemistry, protein characterization, and I won't go through all the technologies, but there are technologies that can be applied to control the process and to understand it step by step by step. And that's exactly what we did. And so in the early 1980s, I was joined by this gentleman, Eric Fries, who came to me from my first postdoctoral fellow, actually, from Uppsala in Sweden, and very well trained in cell biology. At that point, I knew more biochemistry than cell biology. And we did succeed in reproducing this process in a cell-free extract. It took several years. There was a lot of failure on the way. And actually, this is the homogenizer that we used in the very first successful experiments to disrupt the cells. I gave it to Eric as a going-away present when he left the lab. It says this homogenizer, you can't read it, was first used to reconstitute intracellular transport in cell-free extracts, and it has the date of the experiment. And Eric actually provided that now to the Nobel Museum. So it sits in they ask you to donate an artifact to the Nobel Museum of the work, and that actually now sits in the Nobel Museum rather than collecting dust on his desk. So I will always be grateful to Eric. And after several additional years, we learned how to quantify these cell-free transport reactions. And again, this is, it's not important to go through all the details here, but I will say that what we've reconstituted is trafficking into and within the Golgi. So even within the Golgi membranes, there are vesicles that butt off, similar to what you saw, and rejoin to carry proteins across this organelle. And we came up with a very simple biochemical radioactive assay that measures it, an extremely strong signal over a very low background. And any of you who do experiments know that that's what you need in order to analyze any process effectively. And we soon found that there were, soon now being in the 10th year of the project, that there were two chemicals that were very specific inhibitors of this process. Now, the reason inhibitors or poisons are important in biochemistry is they provide a way to understand the intermediate stages. So let's just imagine that this is the beginning of the reaction that we've reconstituted, and this is the end. And at the beginning, we have one membrane and a vesicle buds off in our mind, but we haven't yet shown that a vesicle buds off, right? We're just imagining it does, okay? A vesicle buds off, and now it travels over to the next organelle, all in the test tube, and now it fuses. And when it fuses, it gives it the signal in the assay, the radioactive incorporation. If you have a drug that blocks right here at a very specific place, well, then you will continue to produce the vesicles, let's say, and they will accumulate, but they will no longer, let's say, stick to the target membrane or fuse. And so now, if you look in the electron microscope, you should be able to, the test tube should be filled with these intermediates that were not there before, but were produced in the test tube. Does that make sense? And so the first of these inhibitors, which is a non-hydrolyzable analog of the cellular energy source, GTP, gave a remarkable effect that Lelio Orci, my collaborator, and I at the University of Geneva, a very famous electron microscopist, found. And it accumulated a test tube full of these vesicles. Isn't that amazing? These vesicles were not there before. They had never been seen in a living cell. They'd been missed, okay, even by the great electron microscopists like Palade. And we were able to, but when we blocked transport with this non-hydrolyzable GTP, the membranes, the donor membranes as we call them, were converted to, in very large measure, to these vesicles, which we could then purify. And now we had a test tube full of these vesicles. So these vesicles are now implicated in the transport process. Why? Because when you block transport, they accumulate. Now, if that's correct, we should be able to add those vesicles back to the target membrane. So the vesicle comes from compartment A, and now I want to see, does it go to compartment B? How do I test that? Well, in the test tube, I produce vesicles from compartment A. I isolate them and separate them. You can't, and purify them. That's something you could never do in a living cell. All you can do is watch it. And then I add those vesicles, those pure vesicles that you see here, the transport vesicles, to compartment B, again in the test tube. And now I check, are they intermediates? They accumulated when transport was blocked, but if I now add them to the target membrane, do they go and fuse and deliver their cargo? And the answer is they do. And so that proved that these are the transport intermediates. Now, when you look at them in the electron microscope, and again, I'm sorry, Fred, with the lights. Can we do something about the lights? If we could just turn this off. Thank you. If we leave it off, it's even better. Can you see now that the wall of these vesicles, which carry the cargo protein, have a sort of thick coating material? Can you see that? Can you see the stuff on the outside of it? That's called a coat. That coat provides the mechanism to form the vesicle. And with these vesicles in hand, I won't have time to describe it tonight, we learned how the vesicle is sculpted by this coat. The coat is essentially a protein machine that it assembles into a regular polyhedron, much like a Buckminster Fuller or a geodesic dome. But it does it on the surface of the membrane, and as it does it and it grows bigger, it is round and out pops a vesicle. Okay? Now, as it does that, it incorporates this molecule GTP into the coat as part of the assembly mechanism. And later, that GTP is broken down to GDP, an inorganic phosphate, and that causes the coat to be released. So now the vesicle inside can fuse. And the reason that this non-hydrolyzable analog inhibited is it inhibited the removal of the coat because the GTP was chemically not hydrolyzable. Okay? Now, the other drug that we used, or chemical, was called NEM, or N-ethylmaleimide. And again, the chemical structure of this doesn't matter, but it is a reagent that binds to certain groups within certain proteins. Not every protein reacts. But when it binds, it can inhibit those proteins. And it turns out it inhibits the process of membrane fusion. So now when we look in the electron microscope, we also see lots of vesicles accumulating, but they no longer have the coats. Because the coated vesicles have formed, the coat came off, and now the process of fusion was somehow blocked by this drug. We were able to purify from the cell extracts a single protein that could repair the damage caused by this drug. And we called that Nem Sensitive Factor, or NSF. And NSF was purified, and it turned out it's a protein that exists free in solution in the cell, in the cytosol. So although it's important for membrane fusion in the cell, it isn't actually fusing the membranes. We expected the proteins to fuse the membranes and target the vesicles to be in the membranes themselves in order to have that kind of built-in specificity. Okay? So that was part of the answer, but not the whole answer. We then found that NSF could bind to the membranes. So we mixed it with the membranes and it binds. But in order to bind, it needed an additional protein that we were able to purify also from the cell extract based on the ability to bind NSF. And we called that soluble NSF attachment protein, or SNAP. Okay? So NSF binds to SNAP, SNAP binds to the membrane. Neither one is directly involved in fusion because they're both in the water. So presumably there's some protein in the membrane that we now want to find that's important for fusion. Does that make sense? And that protein would have to bind the SNAP protein because SNAP protein binds to the membrane first and NSF binds after. So we called that the SNAP receptor. We didn't know what it was, but we gave it a name, SNAP receptor. And the SNAP receptor has an abbreviation called SNARE, SNAP receptor. And at that time, happily, I was joined by Thomas Südhof, who came to the laboratory from Munich, where he was extremely well trained by Walter Neupert in protein chemistry. And Thomas was able to purify these SNAP receptors in an experiment that's outlined here. This was a very exciting experiment to the community when we published it. It was actually the most cited paper in all of science, physics, chemistry, biology, and also clinical research that year. It was, of course, completely forgotten the year afterwards and attacked viciously for about 7 or 8 years afterwards, and thus challenging us to establish our interpretation. And that was a good thing. Now, what Thomas did was he took extracts of membranes, mixed them with pure NSF and SNAP protein, and used this hook essentially as bait to pull out whatever bound to these proteins. And we imagined that was the SNAP receptor. And because it was an integral membrane protein, integral to the membrane, we thought that probably means that it is more likely to be directly involved in fusion. Now, to understand what he found, you have to understand where this membrane came from. Okay? The membrane came from brain. So we took cow braids and we ground them up and we isolated the total particulate fraction in a centrifuge, the sediments from the cow brains. And that has all of the membrane proteins in the whole brain. And then out of that, we were able to pull just 3 proteins, just 3 out of those many, many thousands of proteins that had the ability to bind to NSF and SNAP. Okay? And those proteins are the snare proteins. Now, it's important to understand why we use brain. Okay? not because we expected to solve the problem of synaptic transmission, which we did, by the way, to a certain extent. But the reason that we used brain is it simply had the highest binding capacity of all the organs in the body for SNAP proteins. So it was just for purely practical reasons. Now the reason I find that amusing is that when I was a medical student at Harvard and I left medicine to do research, I applied to 2 PhD programs. One, neuroscience, and I wanted to study the synapse and understand synaptic transmission. The other, biochemistry. I was declined by the neuroscience department. I was accepted by the biochemistry department. Therefore, I did not work on synaptic biochemistry. I did not study synaptic transmission. Other people did. I instead went the path that I went. And so to find that our work accidentally, essentially en passant, solved the problem of synaptic vesicle release was really a great lesson, for all of you who are young scientists. Don't sweat too much what you're working on, because you never know where it's going to lead. Now, what Thomas found, and this is the actual notes from sequencing these bands, he found 3 proteins that had already been identified by other colleagues in the field who were working specifically on synapses. And they had painstakingly characterized the dozens of different proteins in the synaptic vesicles that store the neurotransmitter in the brain. And they had characterized several of these proteins, but nobody really knew what the different proteins did. They were known to be there and probably important for synaptic transmission, but not exactly how. But when we found that these proteins that these proteins resided in the synaptic vesicle and the plasma membrane, and that they formed a partnership to each other that we called the Sneer complex, out of this one result came a major idea that we call the Sneer Hypothesis. And that is that these receptors for the SNAP proteins actually bind each other between the 2 membranes, and in binding each other, they provide the driving force for fusion, and at the same time, their specific partnerships, which we proposed occur, provide the specificity for the vesicle to choose its target. And so we called the protein that emerged from the synaptic vesicle a vesicle snare, and the snare proteins or SNAP receptors emerging from the plasma membrane a target membrane snare, or you might call it a plasma membrane snare, but we called it a T-snare because we generalized this idea immediately, because at that time we were already thinking about sorting of proteins and the cargo delivery process. And I think you can now see what our way of thinking must have been, that we imagined that each vesicle has a similar V-snare on it, similar to the synaptic vesicle, but it's different from one vesicle type to the other. So each carrier is marked by a unique V-snare. And each target membrane, B, C, D, E, is marked by a unique target snare. And that there would be a rule in which V1 binds to T1, V2 binds to T2, a sort of code in which the specificity of memory of the delivery process is encoded in the partnership of the V and T snares. So we call that the Sneer hypothesis, with the further idea that when that partnership forms, the fusion process occurs. Now I'd like to fast forward many years, and I see I have about 10 minutes left. And so I'll try to go through this part rather more quickly. We now have a much clearer idea of the physics of the fusion process. The V and the T snares are alpha helix forming proteins. I'm now going to get more technical. They form a 4-helix bundle. That 4-helix bundle of helices that are packed knobs in whole, very much like keratin in hair, is extremely stable. And it forms, this 4-helix bundle forms between 2 membranes, and it forms by assembling progressively in one direction, towards the membrane. And there's an interesting situation that results. Because this 4-helix bundle extends right the way through the membrane, okay, it can assemble or zipper up, this is what we call a snare pin or a trans-snare complex. And as it begins to assemble, it brings the membranes together. It therefore forces the membranes together, but it can't fully assemble until the 2 membrane anchors are in the same membrane, because if they're in different membranes, they can't bind each other. And so this creates two exclusive states: a snare complex that is trying to assemble but can't complete its assembly until the membranes fuse, or the membranes have fused and the snare complex has completed its assembly. There is no other possibility. As a result, the assembly or the folding of this protein is thermodynamically coupled or linked, as a biochemist or a biophysicist would say, to the fusion of the two membranes. What NSF and SNAP proteins do is they act on the assembled snare complex after fusion, and they utilize the energy of ATP to disrupt the snare complex into V and T snares so they can be used once again for another round of fusion. So they turn out to be involved in recycling the snares so that they can be used again, and they are not involved in the actual fusion process directly as we initially suspected. Now, here's a sort of simulation of what this looks like to the best of our current understanding. And a key point is that the T snare contributes 3 of the 4 helices, and the V snare contributes the 4th helix to the 4-helix bundle, as Reinhard Jahn, my colleague in Goodington, and Axel Brunger at Stanford discovered in a collaboration some years ago. And the important point is that the V snare is unfolded by itself. It's a random coil of polypeptide. So that when the 4-helix bundle forms, a lot of energy is released, and that energy is used to do work on the lipid bilayers. And the assembly process is templated by the T snare. The V snare first begins to assemble at one end, and it assembles progressively. And I think you can see that as you take this long random coil and convert it progressively into a condensed helix, that this residual length gets shorter and shorter. As the V snare turns into a helix, this random polypeptide is now condensed, and the membranes are pulled together. Okay, and this happens progressively. And at a certain point, as the snares continue to assemble by this zippering process, the water that is in between the two membranes has to be squeezed out in order to get the two membranes into contact. And Fred and I are developing a hypothesis that suggests that when this occurs to an extreme extent, the water is actually under pressure. We have estimates of the pressure, and we don't have exact estimates, but in various ways we do have estimates, and we think it could be as high as 100 times atmospheric pressure in there. So it's very pressured. And when lipid bilayers are placed under extreme pressures, they become destabilized. They no longer exist in the bilayer, and they actually convert into non-bilayer structures, very much like soap molecules form. And as a result, the layers of this membrane become disrupted, we believe, in one or another way, forced by this pressure to join first the outermost layers, then the innermost layers. And when both layers of the lipid bilayer have joined, this opens a hole called the fusion pore. And if this were a synaptic vesicle, the neurotransmitter would already be released, which is one of the reasons why it can come out so quickly. But then this fusion pore or hole will expand, and as it does, the vesicle will be absorbed into the target membrane, thereby allowing larger molecules like proteins and secretions to be delivered. Okay? So this is what I meant before when I said the delivery truck forms at the starting membrane or compartment A, and is consumed at compartment B, and it lives only transiently. It's really a very elegant solution. That way the cell doesn't waste its energy in having large numbers of carriers when it's not using them. Okay? We don't need to have a big parking lot for all those Federal Express trucks. You just make them when you need them, and you get rid of them when they do their job. What could be nicer than that? Now, in case anyone doesn't understand this as well as I'd like you to, let's picture a hairpin. And let me just pause this at a certain point. Good. Now, if you take a hairpin, it takes a lot of energy, a lot of work, to open the pin, right? That makes sense? And if you let go of the hairpin, what happens? Goes back, right? Well, I'm going to make an analogy that this hairpin is the equivalent of a snare pin. Okay? Why do I make that analogy? Let's imagine that you open the hairpin, as in the earlier part of the video, and you insert one end into a rubber ball and the other end into another rubber ball. And now you let go of the pin. The pin closes down, but it can't close completely, right? What it will do is push these two pins together, push these two balls together, right? So this is very much like a zippering snare complex, which zippers towards the membrane, and each of these balls is like a membrane-enclosed vesicle. Now, if it's a solid or largely solid ball, obviously the two are never going to merge. But what if the ball has a more liquid-like character, like a biological membrane does, or a soap bubble does? And what if the force that's being exerted by this pin exceeds the surface tension of these two spheres? What will happen? Well, you know what will happen. That's what will happen. Okay? So that's the principle of membrane fusion. It's just that simple, except cells have used snare pins instead of hairpins. And there's another very important difference. A hairpin, this is the V half of the snare, the V half of the hairpin, and the T half of the hairpin. Now in the cell, V and T are separate because they have to come from separate places. V marks the vesicle. It joins with T to then assemble the pin. Whereas here, in this analogy, V and T are linked together. But other than that, it's actually quite an accurate analogy. Time-wise?
We have to stop soon.
Okay. So I won't show you the experimental evidence for this, where we've taken single molecules and we're able to actually melt them and watch them reform, to actually watch the physics of this. But I would like to leave you with one idea in the last minute or two, which is an idea that we're currently developing. How is it that the synaptic vesicle can release its content so quickly? Remember I told you that within 1 millisecond the synaptic of the electrical impulse, the synaptic vesicle releases neurotransmitter. Every other fusion process in the cell, like the secretion of insulin and so on, takes seconds to minutes. Why? How can it be so much faster here using the very same snare proteins that everybody else uses? Even worse, snare proteins automatically fuse, right? They just automatically bind each other and they will automatically fuse. And they automatically fuse every place else in the cell. But here, you can't have fusion occurring automatically because your neurotransmitter would be released all the time. And we'd be really stupid, okay? Because we'd be transmitting information when there was no information. That's how stupid we would be, okay? So how does that work? Well, actually, we don't yet know how it works. So this is the sort of frontier in the field. But what we do know is that there's a protein called synaptotagmin. It's a horrible name. But what it does, it was discovered by Thomas Südhof, with whom one of the other recipients of the Nobel Prize in Medicine this year. And it actually sits on the vesicle. And it's a calcium ion binding protein. And when the electrical impulse reaches the nerve membrane here, this opens up specific channels that emit calcium from the outside of the cell to the inside, where it normally is not present in meaningful amounts. That calcium is picked up by synaptotagmin, and in one or another way, synaptotagmin activates the snares. Now, in fact, what synaptotagmin does is it prevents the snares from fusing, and that inhibition or clamping is removed when it binds calcium. So that's how it can fuse so quickly, because it holds the snares as snare pins when they're already half assembled at the very last millisecond. So the question then is, how does synaptotagmin block fusion, and how is it released? And we do not have a firm answer to that, but we have a new insight that comes from the work primarily of two postdoctoral fellows in the lab currently, Jing Wang and Oscar Bello, and in collaboration with another professor at Yale, Chuck Sindelar, and also very much involving Fred and his group here. And what we discovered is that synaptotagmin is organized to make rings. And we don't know for sure, but we think these rings act like a spacer, sort of like a washer or an O-ring, for those of you who make machines, that actually sit between the synaptic vesicle and the plasma membrane. And in so doing, they simply prevent these two membranes from fusing. And then when calcium binds to synaptotagmin, the rings seem to dissociate. And here's an example of an electron micrograph of these rings. And these rings come apart when you add calcium. Here you see the rings. Now you don't. You can quantify it. The rings go away. And if you have a mutant of synaptotagmin that no longer allows synaptic transmission in an animal because it no longer responds to calcium, the rings no longer come apart. In fact, they're more stable. So that's our present thinking, and you can imagine that this is very exciting, but also a difficult proposition to prove. So that's the current frontier. Obviously, many people have contributed to this project over the years. This is the lab as it was when the day the Nobel Prize was announced. You see people look pretty happy, most of them anyway. There's Joy, my wife, who's here tonight. I look pretty happy. Lena looks pretty happy. I don't know what's wrong with Intech. He just didn't seem very happy that day. Anyway, so I thank you all very much, and if there's any time for questions, I'm happy to take them, but if there aren't, we can talk afterwards. So thank you.